Hi everyone, Dr. Mike here. It's important to understand that before a sperm can fertilise an egg, which we call an oocyte, that sperm must undergo two really important events. The first of which is called capacitation, and the other is called the acrosome reaction. Without these two events, fertilisation cannot occur. And that is the focus of today's lecture. So to begin, let's take a look at where we are situated in regards to the female reproductive cycle and the male reproductive cycle. So in regards to the male reproductive cycle, sperm has been produced, sperm has matured, and that sperm has travelled through the male reproductive tract and has been ejaculated within semen into the vagina of the female. So what we've got here is an overview of the female reproductive tract, where we've got the vagina here, we've got the cervix here, we've got the uterus here, and we've got the uterine tubes, also known as the fallopian tubes here, and here we have the two ovaries. So in regards to where we're at in the whole process, sperm has been deposited within the vagina of the female. Now remember, sperm is deposited in semen, and you're going to have around about 500 million sperm, approximately, deposited within this semen. All right, that's where we're at in that process. In regards to the female, so remember, we're trying to talk about fertilization, the process of fertilization. So what we need to have occurred is that an oocyte, or an egg, right, which we know is surrounded by a certain group of cells called granulosa and theca cells, that needs to have ovulated. So ovulation needs to have occurred. So that means day 14 and beyond, right? So day 14 is around about when ovulation occurs. And so now we've got an oocyte, which has been taken up by these fimbriae and is sitting at the ampulla. So around about here. And it's surrounded by a couple of granulosa cells. So this is some point after day 14. Now remember, the sperm must travel from the vagina, through the cervix, through the uterus, through the correct uterine tube to get to the ampulla of the uterine tube where it can fertilise the oocyte. So the window which individuals have in order for fertilisation to occur, so think about this. We need to understand how long does this oocyte last, how long does the sperm last. So generally speaking, the sperm will last about 48 hours and the oocyte will last for around about 48 hours. However, that oocyte or egg won't really last much longer than 48 hours. However, the sperm can last up to 5 days. Up to 5 days. Now that's not common, but it can. And the reason why is because the sperm can actually sit within little folds of the cervix and folds of the uterus and remain there for 5 days. Okay? So that means that if we take the reproductive cycle, right, which we know is day 0 to day 28, right, and then ovulation is happening at around about day 14, that if we have ovulation happening at day 14 and sperm can last, let's say on average 3 to 4 days, that means that you can have sex at around about from day 12, right? Because then you've got day 12, 13, 14. And remember, the oocyte won't last for more than 48 hours after ovulation of day 14. So day 15, day 16. So day 16 is pretty much the last day, right? The last day that sex can occur. And day 12 is probably the earliest that sex can occur. So here we've got the fertilization window, right? So from around about day 12, that's probably pushing it in regards to early. Day 13, probably more so. But between day 12 to day 16 is that fertilization window, simply because of how long the sperm can last and the oocyte after ovulation. So let's think about this, because we haven't spoken about the two important points of capacitation and the acrosome reaction yet, but that's fine. Sperm is sitting within semen. So remember, semen is coming from the seminal vesicles, it's coming from the prostate, it's coming from the bulbourethral glands, and there's some testicular fluid produced in the testes as well. The semen contains a whole bunch of stuff, right? So if we have a look, I'll just quickly wipe this off. We've spoken about that fertilization window. if we have a look very quickly, we know that semen contains a whole bunch of things, right? Semen contains fructose and citric acid, and this can be used as energy substrates, right? It contains prostaglandins. These prostaglandins are important because it aids in the motility of the sperm, but it also helps break through some of the mucus of the cervix and irritate the uterus and uterine tubes to help reverse peristalsis, to push that sperm through. So it contains prostaglandins. It contains coagulants, if that's spelled properly, and things like fibrinogen. This is important because it allows for the semen to coagulate once it's deposited in the vagina, but that doesn't last very long, maybe 15 minutes or so before it starts to break down. It contains a whole bunch of stuff, but something really important that it contains are a bunch of proteins and carbohydrates. And they're there for various reasons. Some are functional, some are receptors, some are nutrients and things like that. Now, think about this. If the sperm is embedded in the semen, let's draw up a sperm, right? So let's draw up a sperm, a giant sperm here. There's the head of the sperm. There's what we'll call the mid-piece. And here is the tail called the flagella, right? So what we need to understand is we've got the head, we've got the mid-piece, we've got the flagella, which is the tail. This tail can obviously swing in a whip-like fashion. The mid-piece is just packed with mitochondria, just packed with it. So you've got all this mitochondria inside the mid-piece. Why is this important? Because the mitochondria is going to produce the energy that allows for that tail to start swinging, right? And then in the head piece, we've got the DNA. So we've got the genetic material. But importantly, sperm know that they don't have any arms or legs to help push their way through this process. They've just got that head. So of course, they wear a crash helmet. And this crash helmet is called the acrosome. Now, the acrosome is just an extension of the Golgi apparatus. And remember, the Golgi apparatus packages things up. So inside the acrosome, we have packaged enzymes. Keep that in mind. So we've got a whole bunch of these packaged enzymes within the acrosome. All right. I'll label that as enzymes. And I'll tell you exactly what those enzymes are shortly. So we've got this crash helmet on the acrosome. Now, because the sperm is in the semen and the semen contains all these proteins and carbohydrates, the head of the sperm is covered in proteins and carbohydrates, right? Because it's embedded in the semen. That's the first thing. So the head is covered in proteins and carbs, right? Second thing is that the membrane of the acrosome is filled with cholesterol, So you've got all this cholesterol present within the membrane here. Now, we need to go back to basic biology, right? Basic cell biology. Why do we have cholesterol embedded in the membranes of our cells? There's many reasons. Sometimes act as receptors, sometimes act as functional subunits. But one of the most important reasons why we have cholesterol embedded in the membranes is because cholesterol keeps the membrane rigid. Remember, our membranes are made up of phospholipid bilayers. They're lipids. It's like oil. If we didn't have cholesterol embedded in our cell membranes, it would have the consistency of oil in a pan. Just fall apart, right? It becomes very weak. So embedding cholesterol into the membranes of our cells, and the sperm is a cell, right, creates it a more rigid and solid membrane, right? So we've got cholesterol embedded here. Now, where does this cholesterol come from? This cholesterol, which embeds within the acrosomal membrane, there's heaps of it, right? More than you probably find in any other cell, because that sperm, when it's made within the seminiferous tubules of our testes, so remember, you've got our testes, need to find space to draw this up. Within our testes, where we make our sperm, we've got, I can probably wipe this off now, what do you reckon? So if I draw up a seminiferous tubule, so here's a testy, right? Here's the epididymis. We're going to have a look inside and we're going to see that we've got seminiferous tubules. We've got a whole bunch of these tubules which has a hollow inside that we call the lumen, right? That's where the sperm will move through. But sperm is produced on the outside here in spermatogonia, which is stem cells, right? And they start to move inward. And as they do so, they mature into spermatids. Now, the process of taking these stem cells, right, to turn into sperm, we need testosterone. And testosterone is produced by these cells that just sit on the outside here, which we call Leydig cells. So this is probably all stuff you've gone through before, but it's good recap. These Leydig cells produce testosterone. Now, they produce testosterone from cholesterol. So remember that cholesterol turns into progesterone. That's an important hormone, particularly female reproductive system, which then turns into androgens. Now, androgens include testosterone, right? And DHEA and so forth. And if we wanted to go to completion, androgens or testosterone turn into estrogen, or the estrogens, such as estradiol and estriol, right? So cholesterol ultimately produces all of these important hormones, including testosterone. So that means, the point I'm trying to get across here is the seminiferous tubule is filled with cholesterol, which means this is where the sperm get all this cholesterol embedded in their membrane. So what have we got? We've got sperm that's been deposited in semen in the vagina after copulation, so after sex, and this sperm has proteins and carbs surrounding its head, and it's also got cholesterol embedded in the membrane. Now, here's a really interesting point. Once that sperm's been deposited, it must travel through the vagina, through the cervix, through the uterus, through the uterine tube to get to the oocyte here at the ampulla. How long does that take? Well, if we were to take the fastest sperm, this sperm will take 5 minutes to get from the vagina to the ampulla. 5 minutes. This is Formula 1 sperm. This is Daniel Ricciardo sperm. That sounded horrible. I'll take that back. Let's just say it's Formula 1 sperm, right? So it can get there in 5 minutes. But here's the important point. Even if the sperm does get through the vagina, does get through the cervix, does get through the uterus, does get through the uterine tube, and makes it here to the ampulla, and let's just say it does it in 5 minutes, can it fertilize that egg? The answer is no, it can't. Because what we need to do is the sperm needs to undergo a process of capacitation. This is where we now talk about capacitation. Capacitation is the cleaning. We need to get rid of these proteins and carbs, and we need to get rid of this cholesterol. That's capacitation. It needs to be cleaned. And this process of capacitation, it needs to undergo capacitation. This takes 7 to 10 hours. So even if the sperm gets there in 5 minutes, it still needs 7 to 10 hours to clean its head. That's what happens. It's basically cleaning its head. Now, how does this happen? Well, as it moves through the female reproductive tract, it's going to come across various fluids and enzymes. And these fluids and enzymes, of which there are a whole bunch, and we don't actually know all of them yet, they clean away the head of the sperm. Clean the proteins away. So we've cleaned those proteins off the head, like that. And here's the other thing, it removes those cholesterols as well. Now, if you start to remove cholesterol, right, I'm just going to do it like this. We start to remove cholesterol from the acrosomal membrane here. What does it look like it's done to the acrosomal membrane? Remember what I said the importance of cholesterol was? In a membrane, it makes it rigid. Well, it makes it weaker and unstable. So now we've got this weakened, unstable membrane of the acrosome. as it moves through. So capacitation is the cleaning of the head, getting rid of the proteins and carbohydrates, removing some of those cholesterols, making the membrane weak. Now, when the membrane becomes weak, some things can happen. First thing is receptors that were present, intracellular receptors, can start to move to the surface, right? So now we've got some receptors moving to the surface. That's the first thing. Second thing is because now it's weakened, we now allow for calcium influx. So we've got all this calcium sitting outside that can jump in. Now what this calcium can do is a couple of things. First thing that this calcium can do is it can make its way to the mitochondria, right? And make its way to the flagella, phosphorylate proteins in the flagella, and it stimulates hyperactivity or hypermobility. So calcium influx results in hypermobility of the sperm, which means now the sperm, they've pressed the NOS button, right? And now this sperm starts to really fire its way through. Now, what speed does sperm travel? It's around about 4 millimeters per minute on average, but obviously if it gets there in 5 minutes, it can travel a lot faster than that. Some are a lot slower, most is killed off. Of the 500 million sperm that are deposited in the vagina on average, how many make it here to the oocyte? Around about 500. 1 in a million. So when your mum said to you, darling, you're 1 in a million, not only was she just trying to be kind to you, she was being biologically and scientifically correct. You are indeed 1 in a million. So what we've got from capacitation, clean the head, carbohydrates off, proteins off, cholesterol off, weaken the membrane, calcium influx leading to hypermobility of the sperm, and once the calcium influx has occurred, remember when we spoke about the nervous system? Once the signal is sent down the end of the neuron, calcium enters. And that calcium influx triggers exocytosis, the pushing out of substances. For the neuron, it's neurotransmitters, right? So calcium influx leads to the pushing out of substances. Same thing happens here. These enzymes that are present start to get released. All right, where are we at now in this process? The sperm should be here at the ampulla by this point. Because remember, this process takes 7 to 10 hours. So by that time, the sperm should be at the ampulla. So now we need to talk about this sperm coming into contact with the oocyte and the egg. Capacitance has occurred, right? Capacitance has occurred. Now let's talk about the oocyte. So I'm going to draw up a giant oocyte here. So there's the oocyte shell, right? But we know that there's a space between the oocyte here and the next structure, which we call the zona pellucida. So here's the zona pellucida. I'll write it here. Here's the zona pellucida, and in here is the oocyte or egg. Now remember I said once ovulation occurred, the oocyte still has some granulosa cells around it. And that's exactly what it's got here, is it's got some granulosa cells present. Trying to protect it, trying to nourish it, trying to keep it safe and well. But it means that this sperm needs to get through these granulosa cells. That's the first hurdle it needs to overcome once it gets to the oocyte. How does it get through? Now we're starting to talk about what we call the acrosome reaction. Because once capacitation has occurred, you can see that the acrosome is unstable. Enzymes are being released from the acrosome. These enzymes are called hyaluronidase and acrosin. Now, what are these? Basically, they're enzymes, and we know that because they're both, well, that ends in A's, but acrosin is an enzyme. Hyaluronidase, it breaks up carbohydrates, right? it breaks them up. Acrosome is a protease, so it breaks down proteins. So there's enzymes in the acrosome that break down carbs and proteins. And what do you think these cells are made out of? Carbs and proteins. So the release of these enzymes start to allow for the sperm to carve a path through the granulosa cells until it can get to the zona pellucida. These enzymes will further break down the zona pellucida, but also, it won't break it down completely, to be honest. I should draw it up like this. It will facilitate not only the breaking down, but because this wall's weakened and we've got these receptors, or at least these binding molecules available, the sperm itself can now bind with the zona pellucida. So we've got binding of the sperm with the zona pellucida because we've got these binding proteins that have come to the surface, and so we could just say that we've got these binding proteins here coming to the surface. Brilliant. So now we've got the zona pellucida bound. Here's an interesting point. Once this has now happened, right, and you've weakened the wall of the zona pellucida, just like we weakened the wall here of the acrosome, what happened? Calcium influx. We get calcium influx again. The zona pellucida gets, because there's been a weakening of the walls from these chemicals, calcium influx occurs. And once we get calcium influx, what did I say happens? The exocytosis of stuff. So what we have inside the oocyte, right on the edge here, are these granules called cortical granules. Cortical granules. These cortical granules contain these substances that once the sperm has bound and started to degrade the wall of the zona pellucida, these cortical granules are released into, release it here, into this space here. Now this space is very important because once the granules are released into this space, what it does is it hardens the zona pellucida. It ends up resulting in the zona pellucida hardening. Everywhere else but the part that it's bound to. And that's important. It's just like, the zona pellucida is like the security system of your house. If somebody sneaks through the window and the alarm system goes off, so this sperm has snuck in, the alarm system's gone off, every door, every window, everything just shuts, which means because 500 sperm have made it here, right? This won't be the only sperm that will have bound. There's going to be other sperm that have bound. But once the screen doors have gone down, any receptors that were in the membrane, they just pop off, right? And all the sperm that's there, they pop off, and no more sperm combined. So this is because of these cortical granules releasing their substances. And then what ends up happening is you get a merging of not only the sperm with the zona pellucida, but you get a merging of the sperm itself with the oocyte. So the membranes bind, and once the membranes have bound of the oocyte and the sperm, then we can transfer that genetic material across. And so now we've got the DNA of the sperm here, and that DNA of the sperm from dad is going to bind with the DNA of the oocyte from mum, and we get recombination. Now, if you want to talk about the specifics, once that sperm is bound, that will actually trigger the second stage of meiosis to finish for the oocyte, which means that mum actually doesn't just have one copy of DNA, it's doubled up, and that needs to be popped off. So that ends up jumping out as a polar body, right? But then we've got the subsequent coming together of the DNA. That's the acrosome reaction. So we've spoken about sperm being deposited, the whole process of the sperm traveling through that arduous journey. Spoken about capacitation, cleaning the head of the sperm. It's basically making it capable, capacitation, making it capable for fertilization to occur. And then we spoke about the acrosome reaction occurring, allowing for binding and facilitation of the DNA to be transported into the oocyte. And I hope you enjoyed that very brief-ish video on fertilization. I'm Dr. Mike. Hi everyone, Dr. Mike here. If you enjoyed this video, please hit like and subscribe. We've got hundreds of others just like this. If you want to contact us, please do so on social media. We are on Instagram, Twitter, and TikTok at Dr. Mike Todorovic, at DrMiketodorovic. Speak to you soon.